And then the other thing that I aspirationally hope to be able to encounter is the ability to rapidly respond to something brand new, whether it's a brand new pandemic or, as you mentioned, a brand new attack upon us deliberately by bioterror.
Holy, are you kidding me?
So let me give you a tough one.
Yeah.
Bioengineering. The ability of state actors,
Right.
terrorist actors,
Right.
to actually weaponize influenza or other bioweapons
Right.
to impact populations. We've heard a lot about this.
Right.
Are we close to having bad actors outside of governments that would have the capacity to develop those?
When you think of a bioterrorist, you think of this person or state that's deliberately trying to release something. Historically, the worst bioterrorist has been nature itself. So when you prepare, as we are, for the natural occurrence of a pandemic flu or antimicrobial resistance, the things we work on every day, it's a natural transition into the same type of preparation against something that would be deliberately engineered to be spread. So is it possible that that could happen? Yes. Are we aware of it? Yes. Do we prepare for it? Yes. We prepare every single day for it with the same type of tools that we prepare for a natural evolution. Yeah, please.
Mr. President, I wanted to ask Dr. Fauci, could you address these suggestions or concerns that this virus was somehow man-made, possibly came out of a laboratory in China? You studied this virus. What are the prospects of that?
Yeah. There was a study recently that we can make available to you where a group of highly qualified evolutionary virologists looked at the sequences there and the sequences in bats as they evolve, and the mutations that it took to get to the point where it is now is totally consistent with a jump of a species from an animal to a human. So, I mean, the paper will be available. I don't have the authors right now, but we can make that available to you.
On the other hand, there have been a number of major breaches that have caused large, not existential, but large real risks or potential risks. In Beijing, I'm just listing the ones in Beijing, there was a laboratory accident with SARS about a decade ago, in which there were 2 primary cases who went on to infect 7 others before it was controlled. There were similar events in Taiwan and Singapore, also with SARS virus, but there were no secondary cases in those examples. There was the famous foot and mouth disease release from Purebright that you're aware of if you've been in the UK since 2007, which caused major agricultural consequences. It's an animal disease, so it didn't cause human consequences. And probably the 1977 H1N1 strain that circulated for 22 years following that reemerged because of a laboratory escape, although that is not proven.
1977 H1N1 was an accidental release, no doubt about it. We don't want to see that happen again with a virus like this. What we're trying to do is come to the middle. How to do this research safely, how to do it in a way that furthers all the points we've just talked about, and at the same time, make sure that we don't create our own problems here. And that's why I think we're asking for this pause to think about this and to move forward in a positive, proactive way.
But, now the question becomes, was it a naturally occurring horseshoe bat coronavirus that was isolated in the Wuhan Institute of Virology and then accidentally escaped from the lab by infecting a lab worker? Or was it a virus that the researchers there were working on using gain-of-function technology? Gain-of-function means that you take an existing coronavirus and you make it more infectious and more deadly. Now, why would you ever engage in gain-of-function research? Why would you ever seek to enhance a natural pathogen in this way to make it more deadly and more infectious? Well, the idea of the scientists, when they first discovered that they could do these things easily by using recombinant technology, was this: we're going to create superbugs in the lab, and we're going to figure out how to defeat them. We're going to develop in theory, we're going to develop vaccines, we're going to develop therapeutic treatments, and so when the real superbug comes on the scene, we will be ready for it. Well, that's all well and good, and that's all fun and games until you create a superbug in the lab and it escapes before you have a chance to develop a vaccine or a therapeutic treatment for it. Now, I'm not saying that's what happened in this case, and I'm not saying this is a bioweapon. Quit changing the subject, which is what the other side loves to do.
Exactly.
I'm just saying, was it tinkered on in the lab? And we know that the lab director at the Wuhan Institute of Virology, who was their team leader for virology research, was a woman named Dr. Shi, full Chinese name, Dr. Shi Zhangli, who has been using gain-of-function research for years to create new, more deadly, and more infectious coronaviruses. So Dr. Shi needs to tell us what she was doing and how she was doing it. The Chinese Communist Party needs to release the secret records of the Wuhan Institute of Virology, and then we will all know for sure whether this was a coronavirus in the lab that escaped, whether they were tinkering on it in any way. Now, I'm not naive in all this. I know that China will probably have already destroyed the records, research records of the Wuhan Institute of Virology. Right. They've been destroying records left and right already. They probably already warned Dr. Shi Zhangli that she is not to talk to anyone about her research. They've warned other people. So I don't think we're ever going to have what people are demanding, hard evidence, because the Chinese Communist Party has already destroyed that.
Well, of course. And there's a reason that they bought and paid for the World Health Organization. It's becoming clear.